Okay, welcome again in my online session on medicinal chemistry. This time we'll discuss about antimalarials. This malaria is caused by a protozoas of Plasmodium species. There are 4 different plasmodiums which are involved in this malarial infection, that is Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae and Plasmodium ovale. Out of that, 50% of malaria is caused by falciparum, whereas 40% by vivax and 10% by malaria. So this malaria caused by Plasmodium falciparum is comparatively dangerous than the other species of Plasmodium. See how this Plasmodium enters into the human and how it causes malaria. Let us see the life cycle of malaria parasite. Now here mosquito acts as a vector to carry this Plasmodium species, and it releases that Plasmodium species into the human blood that is getting multiplied into the human and causes this malaria. Let us see how it happens. The sporozoites which are there in the salivary glands of the female Anopheles mosquito, these sporozoites are released into the human blood during its bite, and this are converted into primary seasons in the liver. Then they forms merozoites. These merozoites enters into the blood. These are merozoites in the blood. In blood, they enters into the erythrocytes, where they consume the components of erythrocytes, multiply and develop different merozoites as well as gametocytes. Now, after rupture of these erythrocytes, due to multiplication of these merozoites into the erythrocyte, it releases, after its rupture, it releases the merozoites, male gametes and female gametes. And due to rupture of the erythrocytes, the foreign proteins are released in the body that acts as an antigen that causes chills and fever. So the major symptoms with respect to this malarial infection is chills followed by fever. The chills because of breakdown of the erythrocytes, and the fever because of the antibody response of the body for the antigens or foreign proteins which are released by the breakdown of the erythrocytes. Now these merozoites again invade fresh erythrocytes, thus the erythrocyte count will be decreased. Most of the erythrocytes are broken down. This is called as erythrocytic stage of the infection. Whereas the merozoites which are in the liver, they again enters into the different liver cells. This is called as secondary seasons. So this Plasmodium falciparum particularly don't have this secondary season site, but all 3 remaining Plasmodiums, they have a secondary season phase. Now the released male and female gametocytes enters into the female Anopheles mosquito when it sucks blood from the infected human. Then it converts into zygotes, oocytes and again sporozoites. That is, a sexual multiplication of these parasites takes place in the female Anopheles mosquito, whereas a non-sexual multiplication takes place in the human body. This is how the life cycle of malaria parasite takes place, and this is how the mosquito, in particular female Anopheles mosquito, spreads the malaria in community. Now there are different stages where antimalarials can act. Some antimalarials can act on sporozoites. When mosquito bites and releases sporozoites into the human body, if they are checked or they are killed at its entry only, so this further multiplication may not take place. So those drugs which are killing the sporozoites are called sporozytocides. There are very few drugs acting at this stage. Then schizontocides. When the sporozoites enters into the liver cells and form liver schizonts, the drugs which are acting at this primary schizonts or secondary schizonts which are formed in the liver, or when they are converted into merozoites, enter into the erythrocytes, that is drugs acting on the erythrocytic phase of the infection. These are called merozytocides. Or there are some drugs which act on the male and female gametes which are generated in the erythrocyte and released after breakdown of the erythrocyte. The few drugs are acting here. These are called as gametocytocides. So this is how various antimalarial drugs shows their action in the human body. Then these antimalarials can be chemically classified on the basis of their structure, like cinchonalkaloids, 4-aminoquinolines, diaminopyrimidines, 8-aminoquinolines, sulfonamides and sulfones, sesquiterpene lactones, quinoline methanol analogs, tetracyclines, amino alcohols, managed bases, naphthoquinone and bigonides. So these are different chemical classes of antimalarial drugs, and different drugs coming under these classes are shown here in this chart. And this chemical class or these chemical classes are based upon their structure. Then look at the first class of antimalarial. These are cinchona alkaloids. The main drugs coming under this are quinine and quinidine, or cinchonin and cinchonidine. These are obtained naturally from the cinchona alkaloids, cinchona plant, a bark of the cinchona. Now look at the structure of this. The cinchona alkaloids, this quinine and quinidine, mainly contains this quinolone ring and a quiniquidine ring. This is a quiniquidine ring and this one is a quinolone ring. And this quiniquidine and quinolone ring are bridged by a methanol, the alcohol group. Right? This is how this quinine and quinidine structure is. Now there are different chemical functionalities present in this, like quinolone ring, secondary alcohol group, cyclic amine and alkene. This is a vinyl group. The alkene present is a vinyl group. And this quinolone ring is substituted with methoxy group. So this is a structure of cinchona alkaloids like quinine and quinidine. If you look at the structural requirement for its antimalarial effect, that is SAR, structural activity relationships of this, is the quinolone ring is must for its antimalarial effect. It intercalates the DNA of this parasite. Then a secondary alcohol, look at this. This secondary alcohol function is required for antimalarial effect because it is involved in binding to the DNA of parasite. This quiniquidine ring is not required, not at all required for the antimalarial effect. It is not involved in this activity. Methoxy, when this ring is not involved, even substituents on this, that is vinyl group, is also not required for the antimalarial effect. Methoxy group on this quinoline is also not essential for the antimalarial effect. If this methoxy is replaced by any halo group like chlorine, bromine, iodine or CF3, it may improve the antimalarial effect. So this is what is SAR of cinchona alkaloids. Moving to the next chemical class of cinchona alkaloids, next class of antimalarials is 4-aminoquinolines. This is a quinoline where the fourth position is substituted by amine. So these are 4-aminoquinolines. If you look at the structure of this chloroquine or hydroxychloroquine or amidoquine or santoquine, here you can see that amine is substituted at fourth position of the quinoline ring, and this amine is further substituted with a long chain carbon. If you look at this, the long chain carbon is substituted to impart lipophilicity to the molecule on this fourth amino group. The chloroquine and hydroxychloroquine, the structural difference, you can have the hydroxy function on this side chain. Amidoquine, the aromatic system, again here the alkyl side chain in the santoquine. So these are different, the 4-aminoquinoline analogues which are acting as antimalarials. If you look at the structural requirement for antimalarial action of these 4-aminoquinolines, here the side chain, if it is shorter, if it is of 2 or 3 carbons, or if it is longer, 10 to 12 carbon, it results in enhanced activity versus chloroquine resistant strains of the parasite. The basic amino group is required in the side chain for its activity. It can be secondary or tertiary or even primary for its moderate activity. The quinoline nitrogen is basic and it is required for the activity. Then a 7-chloro is again the required for the activity, even if we replace the 7-chloro with another halo group with lipophilicity, moderate lipophilicity like bromine or iodine or CF3, they also show some moderate kind of antimalarial effect. Then 8-aminoquinolines as antimalarials. This is a quinoline, 8th position of the quinoline, and amine group on the 8th carbon of the quinoline. So it is 8-aminoquinoline analogues: primaquine, pamaquine, pentaquine, and isopentoquine. These are 8-aminoquinolines, and there is a side chain, a carbon side chain substituted on 8th amino group, like the 4th amino in the 4-aminoquinolines. Here the side chain is placed on 8th amino group. The length of this side chain can be varied with variation in activity. If you look at the structural differences in primaquine and pamaquine, here it's a primary amine in the side chain, and this is a tertiary amine. This amine is substituted with 2 ethyl groups. In pentaquine and in isopentoquine, this is a pentane chain, whereas it is isopentane. Likewise, there is a structural difference between these various analogues of 8-aminoquinolines. If you look at the structural requirement for its antimalarial effect, this methoxy substituent increases activity, but also it increases toxicity. If we place methoxy group at 6th position, though it increases activity, the toxicity also increases. Adding phenoxy substituent at position 5 increase activity and reduce its toxicity. Adding methyl substituent at position 4 again increase activity as well as toxicity. Adding a tertiary butyl substituent at position 2 increases activity and reduces toxicity. An ionizable amino group must be present on the aliphatic side chain. A primary amine reduces toxicity, and acylation of amine reduces a metabolic inactivation. Here, a secondary amine is required at 8th position. So, the 8th amino group is required for the antimalarial effect. So these are various structural features of 8 aminoquinolines for their antimalarial effect. Moving to the next class of antimalarials, dihydropyrimidines and sulfonamides. Here the combination of dihydropyrimidine and sulfonamide is used against malaria. The diaminopyrimidine class of compound here is pyrimethamine and a sulfadoxine from sulfonamide class. These two compounds shows effect because these two compounds act at different stages of folate coenzyme biosynthesis in malarial parasite. The similar thing we have discussed under the sulfonamide class of drugs, so no need to waste time here. That sulfa act here by competitively inhibiting PABA, whereas pyrimethamine, like trimethoprim, it inhibit enzyme dihydrofolate reductase and prevent conversion of dihydrofolic acid to tetrahydrofolic acid and further folate coenzymes which are required in different biochemical processes of malaria parasite. So this is how the malaria parasite can be stopped from its multiplication and further infections. The sesquiterpene lactones, these are newer antimalarials like artemisinin, dihydroartemisinin, artemether and artesunate. Look at this artemisinin, if it is hydrogenated, it is dihydroartemisinin. Look at this ketone, this carbonyl will be converted into a dihydroartemisinin. Artemether is oil soluble, whereas artesunate, it's a sodium salt of this and it is water soluble. So this are various analogues of sesquiterpene lactones which are used as antimalarials, and these analogues are highly stereospecific. Their stereospecificity needs to be maintained to show the desired effect. Mefloquine, it is a drug from a quinoline methanol class. Look at this. This is a quinoline and this is methanol. Our typical moiety is present in the quinine, and this methanol is substituted with a heterocycle, forming a mefloquine, showing antimalarial effect. Tetracyclines, the doxycycline can be used under this class against malaria. Tetracyclines, these are tetracyclines because 1, 2, 3 and 4, the 4 rings are fused together to form this tetracycline. Amino alcohols, halfantine and lumefantrine, these are a polycyclic antimalarials. Like this is a phenanthrene moiety. These are a polycyclic antimalarials. Managed basis, it is again a class of polycyclic antimalarial. 3 rings are fused together, pyrinoridine, and it is showing the antimalarial effect. Naphthoquinone. Naphthoquinone is again a stereospecific compound. This is a naphthalene, and the quinone of this naphthalene, atovaquone, this is antimalarial with a naphthoquinone class of compounds. Then bigonides. The progonyl is a class of bigonide. Look at this. This is a gonadine. 2 gonadine molecules are connected to form a bigonides. Further, it goes to cyclization, forming cyclogonyl, that is active metabolite of progonyl. It is like a pyrimidine or aminopyrimidine class of compounds, and it acts similarly as that of dihydropyrimidine class of antimalarials. Now there are some new approaches for the treatment of malaria, like inhibition of non-mevalonate pathway. Now see, this isoprenoids, which are required for different biochemical processes, these are synthesized in humans from mevalonic acid, that is mevalonate pathway. But in parasites, these isoprenoids were synthesized from pyruvic acid, and this pathway, it is called as non-mevalonate pathway. Means this non-mevalonate pathway is not existing in the human, it only exists in the parasites, that is Plasmodium. So this is what is the selectivity of drugs which are acting by this inhibition, by acting on inhibition of this non-mevalonate pathway. If you look at this, there is enzyme DOXP reductoisomerase, which is required for conversion of 1-deoxyxylulose 5-phosphate to this one, that is 2-C-methyl ethernide phosphate. So for this conversion, it needs DOXP reductase or DOXP reductoisomerase. Here is a compound or drug, that is phosmidomycin, which is isolated from the fermentation broth of Streptomyces. It acts by inhibiting this enzyme DOXP reductoisomerase, which results in preventing biosynthesis of isoprenoids, which are required for different biochemical processes in Plasmodium, and thus it selectively inhibits the Plasmodium species by selectively blocking non-mevalonate pathway, which is not existing in humans. Then another new approach for antimalarial drug action, that is glutathione reductase inhibitors. This glutathione reductase is required to hold heme ion into its ferrous state in the erythrocytes, and if it is inhibited, the further multiplication of these Plasmodium species can be prevented. So here chloroquine and naphthoquinone, these two are combined to show this glutathione reductase inhibition activity. This naphthoquinone portion is involved in binding to this glutathione reductase, and chloroquine is required to carry it to the erythrocytes. So this is how the inhibitors of glutathione reductase can show its antimalarial effect. Thank you.